welcome our next speaker, Dr. Bárbara Gomes, if my pronunciation is correct, and feel free to correct me, who is currently a research associate at Cardiff University at UK. And we are talking about a photochemical approach to for multiplexed biofunctionalization of gallium arsenic. Welcome. Dr. Guo, would you mind to unshare your screen?
Hello, good afternoon and good evening to our colleagues in China. My name is Bárbara and I come from Cardiff University. So I'll start with a brief introduction to our research group, and then I'll talk to you about our lab-on-a-chip optical multiplex biosensor and how we are planning to use it for sepsis diagnostics. I'll explain the functionalization strategy we are using for the biosensor, and I'll introduce you to the technique, the surface characterization technique that we use to characterize the gallium arsenide surface during the functionalization process. And I'll show you some results on this and talk to you about the next steps in this project. So our group in Cardiff is based both in the School of Physics and the School of Biosciences. So in the School of Physics, we have Professor Wolfgang Langbein, and in Biosciences, Professor Paula Bori. And the group expertise is in the ultrafast nonlinear coherent optical spectroscopy. This is not what I will be talking about today, but please have a look at our group website and publications if you're interested in this area. Francesco Mazia is a research fellow, and he's the one leading the project that I will be presenting today. So the aim of this project is to develop a lab-on-a-chip optical multiplex biosensor. And by multiplex, I mean we should be able to detect multiple biomarkers in one sample. So this lab-on-a-chip miniaturized device will incorporate a low-power laser, photonic crystal cavities, waveguides, and detector. And we are a team of 4 at the moment working on this project. So Nadia is a PhD student working on the multiphysics modeling for this device. So she's using Console for this. And Lucas is looking at the setup and the fabrication of the device. Obviously, Francesco is leading the project, and my contribution to this project is the functionalization of the surface with multiple bioreceptors. So a lot of studies have been done in gold and silicon for biofunctionalization, but not so much is published about the biofunctionalization of gallium arsenide. So the way this sensor works is we're looking at shifts in the refractive index resonance when a binding event occurs. And we are hoping to be able to detect up to single molecules. We chose sepsis as the first application for this sensor. And this is because sepsis is difficult to spot and the symptoms can be vague. And thousands of people die per year in the UK from sepsis. It is crucial that we have a fast diagnostic so we can treat the patient. And it is widely accepted that we need a wide panel of biomarkers to detect this disease. And there are hundreds of sepsis biomarkers described in the literature. Obviously, we want to use high specific and high sensitive biomarkers. So from proteins to hormones and microRNAs, all of this we can put together in the sensor surface and use it not only as a diagnostic tool for sepsis, but also to see which stage of the disease we are and to get an indication of the prognosis of this disease. So we chose gallium arsenide as the substrate for this optical sensor due to its optoelectronic properties. And now I will tell you how we are planning to functionalize the gallium arsenide with these multiple bioreceptors. So very basically, we start with gallium arsenide surface patterned with different regions here, circular areas. And we want to functionalize this surface with different bioreceptors. So our strategy will consist of covering the surface with a photoreactive cross-linker, and then selectively expose one area of the surface with the ultraviolet light while incubated with a bioreceptor that we want to immobilize. And then we repeat this for different areas of the surface, different regions, so that we end up with multiple bioreceptors on the surface. And I will go into more detail. As I said, there's not much done about biofunctionalization on gallium arsenide. And for this reason, we started. First, we just wanted to demonstrate we could immobilize a protein on gallium arsenide in a covalent manner. And so we started by not using a photochemical approach, just a more simple strategy where we could immobilize one protein on the surface. And we chose a bioaffinity pair that is widely used in the biosciences. This is biotin and avidin. Neutravidin being a simplified form of avidin. So biotin binds to neutravidin. Each neutravidin molecule can bind up to 4 molecules of biotin, and they have a high affinity. We just published this first functionalization strategy, and we used 3 different techniques to look at the surface changes along the process of the functionalization. So we use spectroscopic ellipsometry, X-ray photoelectron spectroscopy, and wettability measurements. So if you want to have a look at this publication, we are very interested in having a very low thickness for the functionalization film. And this is because we're planning to use this as an optical biosensor that makes use of the evanescent wave of light at the gallium arsenide water interface. From this technique, X-ray photoelectron spectroscopy, we were able to determine what was the ratio of biotin to neutravidin on the gallium arsenide surface. And we also looked at optimizing and reducing the non-specific binding that occurs when we are functionalizing a surface with biomolecules. So that was done, and then we moved into the strategy of using light to functionalize different areas of the gallium arsenide surface. And so we demonstrated that we could now immobilize 2 different proteins in different regions of one sample of gallium arsenide. So now a bit more detail about the chemistry we are using for this. We start by creating a self-assembled monolayer, starting from thiol molecules that are normally used in gold biofunctionalization. One of the thiols exhibits carboxyl groups, and the other one hydroxyl groups. We will use these carboxyl groups for further functionalization. And the other thiol with the hydroxyl group is just there as a spacer so that we can control the amount of bioreceptor that we will have on the surface. A common reaction used in the bioscience is this carbonimide cross-linking reaction. So this enables us to create a stable amide bond between 2 molecules, one with a carboxyl group, the other one with an amide group. And this is widely used. And this is the reaction we use to attach the photocross-linker molecule to the carboxyl groups on the surface. Our photocross-linker molecule looks like this. So it has an amide group that will allow us to attach it to the carboxyl group. And it has the photosensitive group called the diazyrine. This diazyrine molecule is activated by long-wave UV light. And when this is activated, a highly reactive carbene intermediate is formed, and we can create stable bonds with nearby functional groups, for example, a protein if it's present at the surface. So here we can see the structure of our surface at this point. So we have the gallium arsenide with the self-assembled monolayer, and now with the photocross-linker attached via the carboxyl groups that we initially created on the surface. We don't expect this long wave UV light to affect the integrity of the biomolecules we are trying to immobilize. So the idea is to selectively expose one area of the sample for further immobilization of the proteins. So how do we prepare these samples? We start with these gallium arsenide wafers that we buy and we cleave into small samples. We have to clean this very thoroughly with different solvents. We have to remove the oxide layer that naturally is created on the top of the gallium arsenide surface when exposed to the atmosphere. And then we create the self-assembled monolayer by incubating the samples with tiles in solution, so the 2 tiles we are using. Then we proceed with the carbonamide chemistry reaction. There is these 2 reagents, CDC and sulfur NHS, to attach the photocross-linker molecule. And finally, we can then selectively expose the surface to UV light while incubating with the protein or other biomolecule that we want to immobilize. So you can see in the bottom scheme. So we cover the surface with, let's say, protein 1, and then we cover half of the sample and we only illuminate the other half, and this is where the protein will attach, and then vice versa on the other side of the sample. So we demonstrated this for 2 different proteins. We wanted to be able to distinguish between neotravidine and another protein, so we decided to tag this other protein with nickel by using a process that is widely used in the biosciences for protein purification. So we take advantage of the complex that is formed between nickel and the histidine groups in the protein, and we have nickel attached to the protein, and this will enable us to distinguish whether the protein on the surface is the nickel one or the neotravidine. So for the surface characterization, we are using X-ray photoelectron spectroscopy. This technique, I don't know if anyone is familiar with it. This is performed under ultra high vacuum and is very surface sensitive. So we're looking at to the top 10 nanometers of the surface. And basically we radiate the sample with soft X-rays, and we looked at the kinetic energy of the photoelectrons that are emitted from the surface. So from this we can we know which elements are present on the surface, but more than that, we know if a certain element is bound to a more or less electronegative element, and we can quantify the not only the quantity of a certain element on the surface, but also the type of bonds that that element is establishing on the surface. There is a small challenge when using XPS for gallium arsenide, and this is there are a lot of other peaks that appear in the XPS scans that are a result of transitions, not from the core level that we are interested on, but from other levels. And these peaks, they are pointed in with the blue arrows. These are broad features, and they overlap the regions of interest when we're looking at proteins. So oxygen, nitrogen, carbon, and sulfur. We will have some extra peaks there that we'll just have to. The data analysis is just a bit more trickier when we're talking about gallium arsenide. So now I'll show you some of these XPS results for the functionalization, starting from the SAM formation and then onto the photocross-linker attachment, and then show you how we selectively immobilize the 2 proteins on the surface. So here we're looking at carbon. The first plot we can see just bare gallium arsenide without an oxide layer, and this blue peak we can see this is not carbon, this is the peak, the Auger peak coming from the substrate. And then if we look at a sample that was exposed to the atmosphere, there we can see some carbon contamination, and we can see again the peak coming from the substrate. I don't know if you can see the just getting the laser. Maybe it's easy. Can you see? Yeah. So then when the self-assembled monolayer is formed on the surface, we can see that different carbon bonds are detected. And most importantly, we can see the carboxyl group present on the surface as we expected. We know that by varying the carboxyl thiol concentration in solution, we can get more or less carboxyl groups on the surface. As I mentioned, this is important if we want to control the amount of biomolecules that we will immobilize on the surface. Then looking at the photocrosslinker molecule attachment, if we look at carbon, nitrogen, and fluorine, so the crosslinker molecule has 3 atoms of fluorine. We can see that the carbon region changes again after the photocrosslinker molecule attachment. We can see a small nitrogen peak appearing. Again, we have the overlapping of the gallium Auger peaks in this region, but we can still see that there is a peak, 2 peaks of nitrogen appearing, and we can see a shy fluorine peak after the photocrosslinker attachment. When we attach neutravidine on the surface after exposure to UV light, again, we can see an increase in nitrogen. So here I'm showing self-assembled monolayers from 2 different concentrations of carboxyl groups on the surface. And we see that for a higher concentration of carboxyl groups on the surface, we have a higher a nitrogen peak as a result of more neutravidine protein immobilized on the surface. And then if we look at the nickel protein, we can see that nickel is only present on the surface when the surface was exposed to UV light. So here at the bottom, no UV light was applied and no nickel was present on the surface, even if we cover the sample with the nickel protein during the same time as, and we couldn't see any nickel present on the surface, which is a good indication that there is no non-specific binding occurring on the surface. And then finally, here we have a sample where we divided the sample in 2. And as I said before, we covered the sample, half of the sample when for the first incubation, and then the other half for UV exposure, so that we could demonstrate that only the areas exposed to UV light were functionalized with the protein that we were interested. So on the top, we have neutravidine attachment with no nickel present, and at the bottom, we have nickel present as expected. So this photochemical approach for biofunctionalization is quite robust, and we are hoping to apply this now to a multitude of bioreceptors, so going from 2 to 10 or more bioreceptors on the surface. The advantage of having a dual component self-assembled monolayer, as I said, is that we can control the density of biomolecules on the surface. We use this nickel tagging technique just to make the distinction when using XPS to distinguish between the 2 proteins. We have a low thickness film, which is of interest for this type of optical biosensor. And by having neutravidine on the surface, this means we can then use it for immobilization of other biomolecules that have a biotin tag. So if you see in this scheme here, for example, now we can use a DNA strand tagged with a biotin molecule, and we can use the strategy that I described to first immobilize neutravidine on the surface. So we can then attach the DNA strand and hopefully be able to detect the complementary microRNA strand that will, for example, be a biomarker for sepsis. And so the next step will be now to performing measurements on the biosensing device after the functionalization. Thank you.
Thanks a lot for the excellent presentation. We have time for one quick question. So yeah, I got one very quick one is excellent work. So like, what's your next step for your main focus of your research based on this excellent work you have got?
So now is moving into sepsis bioreceptors, and obviously, yeah, use the biosensing device to demonstrate the sensing. Yeah, that's we have a lot going on. Yeah,
Excellent.
This is just the start. Yeah.